Hello everyone, welcome back to another video from Shomu's Biology. In this video, we are going to talk about Mycobacterium tuberculosis characteristics, details regarding the Mycobacterium tuberculosis bacteria. So here, this video will help you for your MBBS first year and any other bacteriology examination. So starting with the features, and we'll start with the gram nature of Mycobacterium tuberculosis. The gram nature is acid fast, not typically categorized by the gram staining, but instead we are categorize it based on the acid fast staining. The next important property is the disease that this Mycobacterium tuberculosis caused, and we know that is tuberculosis disease or TB disease. The third important property is the virulence factors. What are the virulence factors? They carry mycolic acid that is present here. What is mycolic acid? It protects against the host defenses and antibiotics. The second important factor is cord factor that is released by Mycobacterium tuberculosis, impairs the phagolysosome maturation in the host defense. The third important factor that they have is ESAT6 and CFP10. Now, both of them are part of the type 7 secretion system of Mycobacterium tuberculosis bacteria, which is involved in the cell entry into the host body. The next, if we talk about the next important parameter that we talk about is incubation period. Now, the incubation period of Mycobacterium tuberculosis are variably, typically between 2 days to 12 weeks. Now, if we talk about the antibiotic susceptibility, now what are the antibiotics it is susceptible to? What are the antibiotic it is susceptible? The list of the antibiotics are isoniazid, rifampin, and ethambutol, and pyrazinamide. These are the 4 antibiotics it is susceptible to. Now, if you want to talk about the antibiotic resistance for Mycobacterium tuberculosis, then what data we have is increasing resistance due to multiple antibiotic treatment which is not done properly, and the resistance it is found against multi-drug resistant tuberculosis known as MDR-TB and extensively drug resistant tuberculosis or XDR-TB. Both variants are found now. Culture method, if you want to talk about the Mycobacterium tuberculosis, grown on specialized media. What kind of media? Lowenstein Jensen media. And we have Middlebrook 7H10, 7H11 agar medium are the medium where we can grow this bacteria. It forms slow growing colonies, take longer time to be cultured. Detection method for Mycobacterium tuberculosis. What is the detection method? There are 3 detection methods we can talk about. The first method is acid fast staining, and simply looking at this bacteria under microscope, which you can see in this image, identifies Mycobacterium based on the staining properties of its nature. The second important detection method that we have here is the culture methods. Isolate the Mycobacterium tuberculosis from clinical samples, that is sputum and tissues. The third important detection method that we use here is nucleic acid amplification tests, that is the GenExpert test is available, like the brand name, that detects Mycobacterium tuberculosis DNA in it. And the fourth one, the last detection method that we want to talk about, are chest x-rays. Okay, reveals the characteristics of lung lesions which is generally formed in pulmonary TB cases. Okay, so it's kind of a not a direct diagnosis but an indirect way to understand if Mycobacterium tuberculosis is present in the chest or not. So that's all regarding the Mycobacterium tuberculosis. I believe you have a clear idea. If you like this video, please hit the like button, share this video with your friends, and subscribe to this channel to get more videos like that in future. Thank you. Bye.